So for a patient, you say, oh, they came in at 2 years, but I noticed that that you missed all the other visits until then. So that's a way of kind of assessing that. It's pretty simple. The other thing that people often do within trials is they have a certain schedule. So usually they're called windows. And the visit should occur within a certain window. So if you have a visit, say, at 8 months or 9 months, say 9 months, that visit should occur plus or minus 1 month. And you can kind of then tally how the visits are occurring. So if you're managing a trial, for example, that's certainly a management tool to say, at each clinic, what proportion of the visits occurred and what proportions of the visits occurred on time. So you can kind of look at that. So those are different things. And here's a picture of one of these bottles. Well, actually, the thing on the right is one of those bottles. You see a little counter on the top. So every time the top is unscrewed, there's a recording of the time, and you can plug that into your computer and download it. So you see, that's pretty high tech. Now, if I were a participant in a trial, this would just challenge me to say, how can I fool the system? Because you say, yeah, they're really watching me. And so you could imagine you could do it by opening up the pill bottle every day and not taking anything out. Or opening up the pill bottle and taking out the pill and giving it to your friend, or whatever. I mean, obviously none of these things are perfect, and in some ways I think a lot of people think that the higher tech you get, that to some extent you're probably not doing yourself a favor. You might be better off just asking people. But they do have these things, and then there's this thing that I saw advertised. It's called the Sequela Drug Compliance Monitor, and it's a wristwatch that prompts medication ingestion and accurately records that medication has been taken and when it's ingested. Now, I don't know how it does all that stuff, but it certainly could prompt you. You could have some kind of wristwatch, I think. Again, maybe in the era of the internet, we might have some possibilities for automatically emailing people. And as I said in the vitamin D trial, where you have less frequent treatments, that's definitely a more plausible kind of thing. But I think some of these things are fun, and everybody's out to make a buck. So, okay, so what are our goals in adherence? Again, the ideal thing would be, in your ideal great study, all participants continue to take the medication, and they take it perfectly, as they're supposed to, throughout the trial, and attend all follow-up visits until the very end. Okay, so that would be the ideal, kind of the ideal setup. But that's usually not the case. And so it's worth thinking about, I think, why you would stray from that perfect case. Why might participants stop medication or an intervention? First of all, side effects, real or perceived. In other words, we told people, for example, in the FIT trial, they were taking alendronate. We warned them that alendronate might cause upper GI problems, a GERD or whatever, And so we found that there were a fair number of people that said, I did have upper GI problems, and I stopped taking the pill. When the trial was all done and we looked at it, we found that there were an equal number of people in the placebo and the alendronate group who stopped. So clearly, perceived side effects can have an effect on this as well. If the regimes are complicated, if they have to take a pill, say, on an empty stomach or at a certain time of day, that could be a barrier to compliance. Now, often what happens is you get people who sign up for a placebo-controlled or some kind of non-active medication-controlled trial, and sometime during the trial they decide they want to take a true medication. And I'll show you a couple of examples of that. Why that might be is, for example, say whatever you're testing, during the course of your trial, someone else comes out with some information saying this is a great drug. Or say a competing medication comes onto the market. And again, I'll show you an example in a minute. So that might be something that happens so that people are worried that they're on the placebo. So the opposite thing is information may come out during a trial, something bad about the medication you're testing, and the people may be worried that they're on the active medication, and so they may want to stop because of that. And the example would be here is, for example, during some of the trials of estrogen, you had these cohort studies coming out all the time saying estrogen increases risk of breast cancer. So you keep hearing bad information. You say, well, I don't know whether I'm taking estrogen or placebo, but I hope I'm not taking estrogen, so I'm just going to stop the treatment. So those are things, of course, you can't control, but they're things that you might want to sort of keep keep aware of, things happening in the news. And sometimes you as a study may notify participants, say there's going to be some information coming out, and this is what it's going to say, and this may or may not affect your participation. For example, maybe a study says that HRT increases the risk of breast cancer, but the dose that you're using in your trial is much lower. So you may want to tell your participants about this information, but say, we're not sure it applies to what you're taking for one reason or another. So some concrete examples, sort of bad adherence days. This is the case I just talked about, the Women's Health Initiative, which was a study of estrogen. And in this case, not only did they have information coming out into the world from other estrogen studies, it turned out after the first year of the WHI, the results that were seen by the DSMB suggested a slight increase in cardiovascular disease for participants on HRT. So what the DSMB said, the investigators had to send out a letter to participants saying, the study is still continuing, but we found this small increase in cardiovascular disease among the women on HRT in the first year. So as you might imagine, if you were in a trial like that, I get this letter, you would be, really think twice about continuing, and a lot of women didn't. So that was a very difficult thing for the study. And then another example, it's kind of a prototype of things that I think happen quite often. There was something called the PROOF trial, which was looking at nasal calcitonin for osteoporosis. And this trial went on between 1994 and 1999. Now, when they started in 94, there were no approved treatments for osteoporosis. But during the trial in 97, alendronate was approved and had a significant amount of marketing for this drug. And the results were actually pretty good. And so a lot of the women in this trial of nasal calcitonin heard about alendronate and said, well, I want to take alendronate. So whether or not they continued in the trial, they were taking another drug that really affected the outcome in the trial. And so in the end, the trial pretty much had a negative, a null effect. And that may have been influenced by the fact that so many of the women started taking alendronate. Now, so if we look sort of more closely and see what's the effect of stopping medication, well, classically, what you think of is that people on the placebo start taking an active medication and become more like the active medication, and the active people stop taking the active medication and start becoming inactive, more like placebo. So you could imagine that the two groups become more similar under that kind of scenario. And therefore, the treatment effect is underestimated and conservative, because if you use your intention to treat, you'll analyze people as if their original randomization is what they took, and that's conservative. So that's comforting. But I think there are a lot of real cases in the examples I just showed you where that classical interpretation may not hold. And the example was that example of the nasal calcitonin, where it wasn't just that people stopped taking the placebo and started taking active, or stopped taking active and started taking placebo, but people stopped either the active or the placebo and took something that was even more active than what was being studied. So in that case, you can't really be sure that your treatment effect is going to be underestimated. You really don't know what's going to happen. So I think, again, sort of emphasizing the importance of maintaining good adherence as much as you can. So what are some of the strategies to increase adherence other than these little freebies I talked about? A lot of that is in the design of the trial. So when pre-randomization, most trials sort of have a sort of trial by fire, so to speak. So they typically have 2 or more screening visits. So you really are kind of assessing whether people are willing to participate in your trial. And a lot of times people will come in one time and they say, oh, this is really inconvenient. I have to take off work, or I have to go on the bus, or I have to drive somewhere, whatever. And so they don't want to do it. And also in pre-randomization, you can ask people if they plan to move. And a lot of times people say, yeah, I'm going to retire in a year and I'm moving to Arizona or Mexico or whatever. So in that case, they probably wouldn't be a good participant. One thing is also during pre-randomization is you can consider something called a run-in period, where it's a trial run of the drug or the treatment. Typically 2 to 4 weeks. Usually that's of a placebo version of the drug, not always. But the value of run-in periods is somewhat controversial. Some people say